So in this video, we are going to continue on with Lab 1, talking about the diversity of life. And in this video, we're going to go over the answers to the correct classifications and the shared characteristics that those organisms have. So this is going to be day 2. So last time we went through and we talked about how to use a dichotomous key. And so what you do for a dichotomous key is that you start from the top and you start with the first group of statements. And so the first group of statements says either organisms are prokaryotic, most are single-celled and lack a nucleus, or organisms are eukaryotic, single or multicellular, and contain a nucleus. And so we said that the correct statement for that was 1B. So this is domain eukarya. Go to number 3. So you skip over number 2 and you go to statement number 3. And so we worked through this until we end up with our classification. And so our classification for group 1 was that it was in domain Eukarya, kingdom Animalia, phylum Nematoda. And the shared characteristics for this is that they were multicellular, they're worms that lack segmentation, and they have round bodies with pointed ends. And so we talked about some examples. And so that is just to review what was on group number 1. So now, as we walk through this, we're going to go not by the numbers that they were placed in, but instead we're going to go through it by groupings. And so the first group of organisms that we're going to talk about belong in kingdom Plantae. And so for shared characteristics for kingdom Plantae, these would be organisms that have a cell wall. And the cell wall in plants is made of what's called cellulose. Cellulose is a polysaccharide. It's many sugars linked together. And again, that's distinct from bacteria, where most bacterial cell walls are made of something called peptidoglycan. Peptidoglycan is a protein sugar mix. But for plants, their cell wall is with cellulose. Another characteristic that plants have in common, chloroplasts. Not all are necessarily photosynthetic, but at least have accessory pigments. And so they have this structure referred to as a chloroplast, and that allows them, in many cases, to be autotrophs, self-feeders. So let's start with the most primitive of the plants. And this is group number 9. And so if you look at the organisms that are on group 9, what you'll notice is if you look at the scale here, notice here's a penny, and here is the plant. The size is very small. These are mosses. And so for the mosses, what they have in common, let's start first with the taxonomy. So you want to think about and look at for group number 9, what did you classify those organisms as? And if you said they were in kingdom Plantae, phylum Bryophyta, you would be correct. These are the bryophytes. These organisms, these are plants. Notice they grow low to the ground, and that's because these plants lack a vascular system. If you think about our vascular system, right? Think of our cardiovascular system. The job of our vascular system is to move blood and food and oxygen throughout our body. Same idea in plants for a vascular system. The purpose of the vascular system in plants is that it allows plants to transport water and sugar long distances. Notice these plants grow low to the ground. They grow low to the ground because they lack a vascular system. They don't have a vascular system. So they don't have the ability to transport waters over long distances. So instead, they have to grow low to the ground. They have to grow in contact or close proximity to water because the way that they get in water and nutrients is by simple diffusion. It simply diffuses into the plant. And so these are the most primitive plants. Again, they're non-vascular. They are also seedless. They do not produce seeds. Instead, they use spores to reproduce. And you'll notice on the slide, it says spores are produced. And so we will talk about reproductive methods in various organisms. And one of the things that we'll talk about is that in mosses, they have what is called the sporophyte generation. And this top portion of the plant has this structure that has spores in it. And those spores are used for reproduction. And so these do not use seeds. The embryos are not contained within a tough seed. Instead, these plants use spores to reproduce. But the big characteristic that they have in common, they have to be small due to a lack of a vascular tissue. Next, we have group number 3. And so for group number 3, this would include things like a tree fern, a maidenhair fern, etc. So these are basically your ferns. Now, if you look at the classification for this one, for number 3, what did you put as your classification? Phylum Pteropsida. So these are our ferns, Pteropsida. The P at the beginning is silent. Now, if you think about ferns, can they grow tall? Answer is yes, they can grow tall. These plants have now evolved to have a vascular system. So they have these vessels, these xylem and phloem vessels that are used to transport water and sugar. And so these plants now evolved to be able to be tall because of the presence of the vascular system. However, these are still not seeded plants. They don't produce seeds. They reproduce with spores. And so on the underside of the leaf, you can see these little brown dots here. These are little packets that are called sori. And within the sori are the spores. And those spores are used for reproduction. So for the ferns, we're starting to see the evolution of a vascular system, but we still don't have seeds. These are still seedless plants. Next, we come to group number 5. Group number 5. So look at your lab to see what you put for group number 5. And what you'll notice on the slide, the organisms that you'll see here, we have a cycad here, we have a ginkgo plant, we have redwoods, juniper, spruce. These are all examples of this type of organism. How did you classify this group of organisms? Answer is, these again are still plants. They're in the gymnosperm category. So these would be our gymnosperms. Now, for our gymnosperms, notice they're tall, right? If you think of a redwood, they're very large, which tells you that they do have a vascular system. Right? So they are vascular plants. But now what you're starting to see in terms of their evolution is that these produce seeds. And they produce seeds in cones. So these plants now have cones. And within the cones, they have what we call naked seeds. They're not enclosed within a fruit. So in phylum Gymnosperm, or in this grouping, the gymnosperms, they have evolved to have seeds. They're still naked seeds, but they do produce seeds. So their shared characteristic, they're vascular, they're seed producing, and they produce cones and naked seeds. Then we come to group number 7. Group number 7. And we will see some examples here. Manzanita flower, mesquite, San Pedro cactus, deerweed, wheat, Canterbury bells, all these various type of plants. Now, what did you classify this as? Answer, these are your angiosperms. These are the angiosperms. And for the angiosperms, notice that these plants can grow tall, right? So they can grow tall, which tells you they do have a vascular system. They do produce seeds, but now seeds are contained within a fruit, right? And so they have their seeds contained in a fruit. Think about what advantage a fruit gives a plant. Plants, right, are stationary. They don't walk to different areas, right? They're stationary. So in angiosperms, the evolution of having a fruit is an advantage because fruits are tasty to animals. Animals come along, they eat the fruit. When they eat the fruit, the seeds go with the animal. Animal moves, goes to a different area, defecates or goes to the bathroom, and out come the seeds, and they're in feces, which can have a lot of nutrients. And now the seeds can germinate. So having seeds that are contained within a fruit allowed for dissemination of the seeds. It helped basically move the seeds. In the gymnosperms, the group we talked about before, those seeds are different. Those seeds are distributed more by wind. And again, you'll see that when we talk about our reproductive methods. You'll see that on the cones, the seeds have a wing, and that wing is helped for seed dispersal. In angiosperms, the seed is contained within a fruit, and the fruit gives the plant an advantage. The other adaptation that angiosperms have, the other shared characteristic, is notice you'll see flowers. These are our flowering plants. What advantage does a flower give a plant? Well, the answer is attracts pollinators. Different types of flowers attract different types of pollinators. It could be bees, it could be beetles, it could be hummingbirds, it could be butterflies, bats even. But the idea is that the flower is used to attract pollinators so that the animal, for example, comes and it lands and it goes to get nectar, and inadvertently it picks up the pollen, and then the bee, for example, will go and land somewhere else and it will pollinate another flower. So flowers have the adaptation in that they help to attract pollinators. So for our angiosperms, these are vascular. They do grow tall. They are seed-producing plants, but their shared characteristic, what makes them distinct from the other groups, is that they produce flowers and a fruit surrounding the seeds. That's their shared characteristic. And so those are our 4 plant groups. Next, we're going to come along to this group. This is group number 11. And what you can see in group number 11, these are our molds, mushroom, and yeast. Molds, mushrooms, and yeast. So what did you classify group 11 as? And if you said kingdom fungi, you would be correct. Kingdom fungi. So again, mushrooms, molds, and yeast. Molds and mushrooms are multicellular. Yeast cells are unicellular. So this group has both multicellular and unicellular organisms. Kingdom fungi, they have a cell wall with chitin. Chitin is also found in some animals in their exoskeleton, for example. But chitin is a polysaccharide. Just like plants use cellulose, which is a polysaccharide, fungi use chitin. And so that gives those fungal cells structure and strength. In terms of the way that the fungi reproduce, they reproduce with spores. In terms of how fungi get food, is that they are heterotrophic. Heterotrophic means other feeders. They have to consume their food in order to survive. Many fungi are what we call saprobes. They feed on dead, decaying matter. So these organisms, again, shared characteristics, cell wall with chitin. They are heterotrophic. Many of them will feed on dead, decaying matter. So some examples here that I put in here for you to see. This one up here, these 2 images, this is Aspergillus niger. It is black mold, and it's a mold that can be found commonly in the walls of houses. And if you look at it under the microscope, it has these hyphae, these filament structures, and then at the top are the spores that are used for reproduction. And so they have these type of spores. They're called conidiospores. They're naked spores, and they're used for reproduction asexually without a partner. This one down here, this green in this microscope picture, this is penicillium. Penicillium, you can guess by the name what penicillium might produce. It produces penicillin. Interesting story about the way that penicillin was discovered. In 1922, a scientist by the name of Alexander Fleming discovered penicillin simply by accident. He was trying to grow bacteria on agar plates, and he ended up with a contamination. And he noticed that where this fuzzy stuff grows, because molds produce this fuzzy appearance because of their hyphae, because of their filaments, Fleming noticed that on the plates, his bacterial plates, where there was this fuzzy stuff, bacteria didn't grow in that area. And so he said, that's interesting. And pretty much that's how penicillin was discovered. It was a very lucky accident. And so that's how penicillin was discovered. It wasn't mass produced until the 40s. but it was discovered in 1922, and it was widely used after. Here, what you're seeing on this picture, again, we have our hyphae. These are the filaments. They have these naked spores. The penicillium looks like little fingers under the microscope. And so that's what their spore structure looks like. And then one that you've probably seen many times, we have our bread mold or rhizopus. And so again, you guys will see this when we talk about our reproductive methods. But rhizopus, for example, can also produce spores, and it produces them within what's called a sporangium. And it's basically this little capsule that contains the spores. And those spores are used to reproduce asexually. So what you're seeing in these pictures, these are asexual types of reproduction, but there are some mold. Rhizopus also does sexual types of reproduction. But so it can do both. So they don't necessarily reproduce just one way or the other. They can reproduce sexually or asexually. We have our mushrooms here. And so these are just some examples. We have some yeast. This is baker's or brewer's yeast. This is what you can buy from the store. And what that basically is that is an organism called Saccharomyces cerevisiae. Saccharomyces cerevisiae, name literally translates to sugar fungus that makes beer. That's what its name means. Saccharomyces cerevisiae means sugar fungus that makes beer. So what that tells you about that organism, it's a single-celled yeast. And this yeast, if grown under what's called anaerobic conditions, meaning without oxygen, if grown anaerobically, the yeast will take sugar and it will break down the sugar and it'll do fermentation. And the products of fermentation are going to be alcohol, so ethanol, hence how it's able to make beer. It also produces CO2 or carbon dioxide. And so if you think about the uses for yeast, beer and wine, that's because they produce alcohol. We also use yeast to make bread. And you might wonder, well, why is yeast used to make bread? Is it because we want alcohol in the bread? No. What we use yeast for in bread is that the yeast helps the dough to rise. Because again, remember that when they do fermentation, they're going to produce carbon dioxide. And that carbon dioxide is going to cause the bread to rise. Now, does that mean that the bread, when you cook it, after you cook it and you eat it, does it contain alcohol? No. The alcohol bakes off in the oven. If you were to eat the dough, yeah. But once you've baked it, the alcohol is going to bake off, and the purpose of the yeast in that case was simply to make the dough rise. So group 11 would be your fungi. These are your molds, your mushrooms, and your yeast. Now we're going to move on and talk about our kingdom Animalia. So everything else that we're going to talk about from here on out are our animals. So if we look at kingdom Animalia, our animals, things the animal cells have in common: 1, no cell wall. So plants have a cell wall with cellulose. Fungi have a cell wall with chitin. Animal cells, no cell walls. They don't have cell walls. Our cells are very flexible because they don't have a rigid cell wall. And the other thing about animals is, do we make our own food? Can I go out in the sun and just lay out in the sun and make my own food? Answer is no. We have to consume our food to survive. Animals are going to be heterotrophic. They have to consume their food in order to survive. So those are the things that animals have in common. No cell wall, all are heterotrophic. And so let's walk through and look at different types of animals. So the first group, the more primitive of the animals, we have these animals here. And when you first look at them, you might not think they're animals because they almost have more of a plant-like appearance. But in fact, these are animals. We have a glass sponge here. We have a California red volcano sponge here. We have this branching tube sponge. But what you'll notice, and this was indicated on the slide for you to kind of help you, is that they have pores. They're covered in pores. And so what did you classify these as? Kingdom Animalia, their phylum Porifera. Porifera, notice, sounds like pores. What these organisms have in common is that they are covered in pores and they lack symmetry. So remember, we talked about different types of symmetry. Some animals, for example, can have bilateral. They can be divided in half. Some have radial symmetry where they could be divided like a pizza. But in this case, the sponges, they lack symmetry. They don't have that symmetry, that ability to divide them equally. They have kind of more of a randomized structure. If we look at the porifera, the pores, another thing that they have in common, they are aquatic. They live in water. And so what happens is that the water enters the body through the pores. So the water comes in through these pores, and they're essentially filter feeders. Water exits out of, goes out of this top hole, and so they just basically filter what comes in. So water goes in through the pores, and that's how they get their food that they eat. But their shared characteristic, they are covered in pores, they lack symmetry, and they are aquatic. They live in water. So next, we have group 6. So we come along to group 6. And so if we look at group 6, we have our corals, we have a purple striped jelly, we have moon jellies, we have sea anemones, we have a box jelly, we have a Portuguese man-o-war. These are all organisms that fall under group 6. And so what did you classify group 6 as? Phylum Cnidaria. Now again, the C is silent. We don't say the C, so Cnidaria. These are the cnidarians. Now, in terms of their shared characteristics, what did these all have in common? And what you should see is that they contain stinging cells, nematocysts, right? Jellies can sting you, sea anemones can sting you, etc. So these contain stinging cells. They have what's called radial symmetry, right? So if you look from the top down of the jellyfish, it's like a pizza. You can cut it in many different directions. So they have radial symmetry. They are aquatic, right? Notice these all live in the water. And they feed by stinging their prey. And so they have these tentacles, and those tentacles have the nematocysts, and that allows them to sting their prey to be able to capture it. And so these would be our cnidarians. Next, we're going to come to our groups of worms. Groups of worms. And so if we look at these worms, what do these worms have in common? What is their taxonomy first? Answer for group number 8, these are Platyhelminthes. The way I remember this one, flat. These are your flatworms. So Platyhelminthes, we have some examples here. These are both 2 types of marine flatworms. This is a tapeworm. This is one that's called Taniosolium. Taniosolium has this head, and it has these little hooklets on it. The head is referred to as a scolex, and the adult tapeworm can attach to the intestine and can reside in the intestine. The way that you get tapeworm is by eating undercooked meat. Taniosolium specifically, this species, is found in pigs. So for this one, it's when you eat undercooked pork, for example, because pork will contain the larval form of the worm in the muscle. And if that meat is not cooked well enough and you then eat it, you're going to end up with the adult worms in the intestine. These adult worms get very large, many feet long. Taniosolium is found in pork. We have Tania saginata, which is found in beef. There are tapeworms in fish. There are many different types of tapeworms that can happen, but these are going to be your flatworms. We have a planarian. This is a freshwater flatworm. We have a fluke, which can be parasitic. We have a hammerhead worm, which is semi-terrestrial. But what these have in common in Platyhelminthes, they are flat and they are unsegmented worms. In terms of their environment, some are aquatic and some live in soil. So it's not like the cnidarians and the porifera where they are aquatic. These actually have some that live on land. In terms of platyhelminthes, some of these are parasitic and others are not. So that is not necessarily something they all have in common, but some of them are. And I just wanted to point this out because this is kind of an interesting thing. So for Taenia solium, the one that's the pork tapeworm. This particular worm, if you eat the larval stage in the muscle, you end up with the adult tapeworms in your intestine. However, if you happen to ingest the egg, so this is the egg under the microscope, this is a Taenia solium egg. If you happen to ingest the egg stage, so not eating the meat, but the egg stage, if you ingest the egg stage, you get something called cysticercosis. And what happens in cysticercosis is that you get these larval forms of the worm in the brain. So all these little holes you can see there in the brain tissue, those are all little sacs that are containing the larval stage of the worm. And that's by ingestion or inhalation of the eggs. Because the larval stage, remember I said for the tapeworms, right, that the larval stage forms in the muscles of animals. Well, yes, if you ingest the eggs, you're going to end up with the larval form in muscle or brain. And so this is where you would find these. How then do you get this? How do you end up with eggs that you ingest, right? If you have the adults in your intestine and it's not from the meat that you eat, how do you get cysticercosis? How do you get the larval stage in the brain? Answer is somebody who is infected with a tapeworm, somebody who has the adult tapeworms in their gut is going to shed eggs in their feces. Now, if somebody is infected with the worm and they go to the bathroom, and let's say they don't wash their hands well, and then they prepare your food, right? They handle your food, and you now eat that food, the eggs from their hands are all over that food. You ingest that egg, and you can end up with cysticercosis. And so I have a little video here just to kind of show you a little bit about cysticercosis because it is pretty interesting to learn about.
Only on Fox, at this time of year, we hear how important hand washing is to prevent the spread of the cold and flu. Tonight, you'll learn how hand washing could prevent a scary brain condition. A Valley mother of 4 is told that she has a brain tumor. But when doctors go in to remove it, they find something else, something living inside her brain. Rosemary Alvarez savors every minute with her kids.
That was a good throw.
Playing ball and laughing. For this mom, each day with them is a gift after coming too close to death last August.
I thought I had the flu. I started feeling tired.
But within days, this soccer coach and PTA president knew it wasn't the flu.
I started feeling numbness in my left arm, blurred vision. I'm still smiling for my kids, but now I can't get out of bed.
2 trips to the ER solved nothing.
CAT scan came back normal, no problem.
But her husband, George, was scared.
She couldn't eat. She could barely walk to the bathroom. So I knew at that point, she was deteriorating really quick.
It all came to a head on the first day of school as their kids aged 9 through 17 headed back to the classroom. Their mom was in an ambulance headed to Barrow Neurological Hospital after an MRI explained those strange symptoms.
It came back. You have a brain tumor. At that exact moment where my heart fell just completely all the way down.
But the news would get worse. Neurosurgeon Dr. Peter Nakaji.
Once we saw the MRI, we realized this is not good. It's something down in her brainstem, which is as deep into the brain as you can be.
George broke the news to the kids.
It was a rough day because it was the first day of school, and they came home with all their stuff from school and all the stuff to sign, and look what I got from my teacher, and where's mom, and what's mom? Well, mom's in the hospital, and this is what happened.
As Rosemary waited for surgery, she worried.
Oh my God, we just canceled our insurance like 3 weeks ago, and now this happens, and mostly, how are we going to pay for this?
And she prayed.
Okay, God, if you're going to take me, but make sure my kids are good.
By the time the surgery got underway, all those PTA moms and neighbors, her kids, they were all praying too.
My girlfriend, she goes, everybody's calling and saying they're praying for you, they're praying for you. Church, they're going to church and they're having the mass, pray for you. And pray. Power of prayer. That's what I told them. Power of prayer.
It worked. Dr. Nakaji was expecting to find a brain tumor. He showed me the video as he operated on Rosemary's brain, carefully working, knowing this is the most dangerous spot in the human body to work next to, the brainstem.
Here's the lump, and at this point it still looks like a brain tumor, and so we're just opening up into the inside.
And then the unexpected. No tumor, instead a worm. Watch as Dr. Nakaji grabs the worm and gently tugs it out of Rosemary's brain. It's still alive. You don't hear him on the tape, but Nakaji actually chuckles.
Which I'm sure is a very strange response for the people in the operating room, but because I was so pleased to know that it wasn't going to be something terrible.
This worm comes from eating undercooked pork, but more frighteningly, it can be spread by people who have the parasite in their bodies and don't wash their hands well after using the bathroom. That is the problem.
And it only takes one person who is spreading it constantly to get a lot of people exposed, and some of those people are going to go on to develop this problem.
It's impossible to pinpoint exactly when Rosemary contracted this brain worm.
Probably she was one of these very unfortunate people who had somebody handle food for her, that she ingested it, and it got to her brain.
What is normally a rare occurrence has become much more common. Dr. Nakaji has removed 5 of them in the past few months, and other doctors at Barrow, they're seeing more of them too.
I think it could be a fluke, but it does concern me that if we see more and more cases, as one person is infected and starts to spread these, it spreads it to other people who become infected, and then they spread it, so that more people are at least at risk.
Meantime, George and Rosemary are stunned, relieved, and a bit creeped out by it all.
It could have ate holes in her brain like a apple.
This whole part of my head, from here to here, it's numb. I just learned to live with it.
Rosemary shows off the scars.
I had to learn to walk, to balance.
But Rosemary says the most important thing she learned is how to really live.
So that was cysticercosis, and so you can see that if you happen to ingest the eggs, again, by somebody not washing their hands and having fecal matter and eggs on their hands, if they prepare your food, you could end up with cysticercosis. And so that was just to give you an example of something that can happen from a platyhelminthes, a flatworm. So next we come along to group 10. And if we look at group 10, we have these earthworms. We have this polychaete, which is a marine worm. We have a leech, which is used for, it sucks blood. And so if we look at these worms, what do these have in common? Or first, let's start with taxonomy. What is their taxonomy? Annelida. These are the annelids. And these are going to be worms. Notice these are worms, but they are segmented worms. And so again, you can see that they have these little ridges. These are the segments. And in these various segments, they might have, each segment might have a heart or whatever structure it might be. But these are basically your segmented worms. In terms of where they live, they're not all aquatic. Some can be aquatic, right? So here's that marine worm, and the leech can be aquatic, but an earthworm can live on land. So they don't have their habitat in common, but what they do have in common is that they're all worms, and they are segmented worms. And so these would be your annelids. For group number 13, this includes an octopus, squid, chitin, a garden snail, abalone, clams, etc. What classification are this group of organisms for group 13? Answer is phylum Mollusca. These are the mollusks. What these have in common is that they have one muscular foot for movement. One muscular foot for movement. In the squid, for example, it's not necessarily a foot, but actually in the head. But if you look at the garden snail, this is the muscular foot for movement. Or in the clams, etc. So what the mollusks have in common is that they have one muscular foot for movement. Then we come to group number 2. Group number 2 has a lot of organisms in it. So we have, here's some examples: a horseshoe crab, a black widow spider, a scorpion, a lobster, a crab, a roly-poly, a beetle, a damselfly, bee and wasp, centipedes. These that can be found at your house, these aren't dangerous. They look a little creepy, but they're not necessarily dangerous. That's a centipede. We have mosquitoes, we have a katydid, we have a butterfly. So what classification did you get for group number 2? Group number 2. This would be phylum Arthropoda, the arthropods. So this is one of the most diverse group of organisms. What they do have in common is that they have an exoskeleton, meaning the skeleton's on the outside, and the external or exoskeleton is made of chitin. Just like fungal cell walls have chitin in them, so do animal, not the cell wall, but the exoskeleton in this case. The other thing that they have in common are their jointed appendages. So if you notice, if you look at their legs, they have jointed appendages. And so that's the other main characteristic that they have in common. That's their shared characteristic. So exoskeleton and jointed appendages. And so this would be your arthropods. Group number 4. Group number 4, phylum Echinodermata, the echinoderms. This includes our sea stars. So here are 2 sea stars. Here is a brittle star. Here is a sea cucumber, sand dollar, a red urchin or a purple urchin. So these are sea urchins. So for Echinodermata, what do they have in common? They do have an endoskeleton. So the skeleton's on the inside. Actually, this hard part that you see on the outside is not an exoskeleton. They do have a skeleton internally, so they have an endoskeleton. They are covered with a spiny skin, and they have radial symmetry. So again, it's like a pizza. You can cut them in different directions to have them be symmetrical. They do start as bilateral symmetry, but they finish with, as adults, the radial symmetry. Shared characteristic for this would also be a water vascular system. They have a water vascular system. And another shared characteristic, these are aquatic, right? They live in water, and they do so. Notice there's even a picture here. Here's the water going in. It has a water vascular system. So next we come to group number 12, so station 12. And so if we look at this group of organisms, there's a lot. There's various types of fish, there are amphibians, there are reptiles, there are mammals, there are birds. What is our classification for this group of organisms? Answer is they are in phylum Chordata, subphylum Vertebrata. These are the vertebrates. And so in phylum Chordata, what they have in common is that they have a notochord. Again, it's a cartilage structure. It's a precursor to a vertebrae. But in subphylum Vertebrata, they actually do have a fully formed vertebrae. They have a dorsal nerve cord. Dorsal means like in the back. They have a postanal tail. In humans, your postanal tail is your tailbone, your coccyx bone. And they have what are called pharyngeal gill slits. And so the shared characteristic for phylum Chordata, notochord. They have a structure called a notochord. That's what these all have in common. And so this concludes our diversity of life lab.